everyone, welcome to the session on genome databases. In this session, we will study about the various kinds of genome databases, their organization and their attributes as well. We would also gain an insight about how to use information retrieval system to obtain data and the need to analyze sequence data. Introduction to biological databases. All living organisms are characterized by the capacity to reproduce and evolve. The genome of an organism is defined as the collection of data within that organism, including the set of genes that encode proteins. In 1995, the complete genome of a free living organism was sequenced for the first time, the bacterium Haemophilus influenza. In the few years since then, the genomes of thousands of organisms have been completely sequenced, ushering in a new era of biological data acquisition and information accessibility. Publicly available data banks now contain billions of nucleotides of DNA sequence data collected from over 260,000 different organisms. 3 publicly accessible databases store large amounts of nucleotide and protein sequence data. GenBank at the National Center for Biotechnology Information (NCBI) of the National Institute of Health (NIH) in Bethesda, the DNA database of Japan at the National Institute of Genetics in Mishima, and the European Molecular Biology Laboratory (EMBL) nucleotide sequence database at the European Bioinformatics Institute (EBI) in Hingston, England. These 3 databases share their sequence data daily. They are coordinated by the International Nucleotide Sequence Database Collaboration (INSDC). GenBank. GenBank is the NIH genetic sequence database, an annotated collection of all publicly available DNA sequences. GenBank is part of the International Nucleotide Sequence Database Collaboration, which comprises the DNA Databank of Japan (DDBJ), the European Molecular Biology Laboratory (EMBL) and GenBank at NCBI. These 3 organizations exchange data on a daily basis. A GenBank release occurs every 2 months and is available from the FTP site. The release notes for the current version of GenBank provide detailed information about the release and notifications of upcoming changes to GenBank. In more than 30 years since its establishment, GenBank has become the most important and most influential database for research in almost all biological fields, whose data are accessed and cited by millions of researchers around the world. GenBank continues to grow at an exponential rate, doubling every 18 months. Release 194, produced in February 2013, contained over 150 billion nucleotide bases in more than 162 million sequences. GenBank is built by direct submissions from individual laboratories as well as from bulk submissions from large scale sequencing centers. Organisms in GenBank. Over 260,000 different species are represented in GenBank, with over 1,000 new species added per month. The number of organisms represented in GenBank is shown in the table. Eukaryotes have a nucleus and are often multicellular, whereas bacteria do not have a nucleus. Archaea are single-celled organisms distinct from eukaryotes and bacteria, which constitute a third major branch of life. Viruses, which contain nucleic acids, DNA or RNA, but can only replicate in a host cell, exist at the borderline of the definition of living organisms. From the table, we can see that the organisms in GenBank constitute mostly of eukaryotes. Of the microbes, there are currently over 25 times more bacteria than archaea represented in GenBank. To help organize the available information, each sequence name in a GenBank record is followed by its data file division and primary accession number. The following codes are used to designate the data file divisions: PRI, which denotes for primate sequences, ROD for rodent sequences, MAM for other mammalian sequences, VRT, other vertebrate sequences, INV, invertebrate sequences, PLN, plant, fungal and algal sequences, BCT, bacterial sequences, VRL, viral sequences, PHG, bacteriophage sequences, SYN, synthetic sequences, UNA, unannotated sequences, EST, EST sequences, PAT, patent sequences, STS, STS sequences or sequence tag sites, GSS, GSS sequences or genome survey sequences, HTG, high throughput genomic sequences, HTC, high throughput cDNA sequences and ENV, environmental sampling sequences.